the slide. Maybe you already read about this type of experiment because it's quite popular for the scientists who work on the biologists. But also I still put here in order to help you know more about the history of the cell research in the 19th century. So this is the experiment they perform to answer the question, where do cells come from, or are all the cells come from the pre-existing cell? So they use the two bottle, the glass bottle, as you see here. So the one bottle is sealed, and the other is open. And in these two bottles, they contain some highly nutrient solutions for the cell, like bacteria grow. And these bottles, boiled. They are boiled. They are heated to the boiling temperature to kill all the microbes present in the solution. And after that, they let the two bottles in the normal temperatures, with the one is open and the other is sealed. So for the time, they observe the trends in the color of the solution in these two bottles or in these two containers. The open one, the color turn darker, and the sealed one, they keep unchanged. So if the seal container is broken or open, the solution changes the color. Okay, so that is the result they receive from the experiment. And why the color change in the solution? Because in each solution they put the indicator, the pH indicator. You know the pH indicator, right? Because when the pH value changes from acidic to basic, so the indicator will change the color. Like the phenol, they change the color from the pink to the yellow if the basic turn to acidic. Okay, the pH changing. So in this open container, if there is a cell present in the solution, so the cell will use the nutrient, and the metabolism products will change the pH in the solution, and they make the indicator change the color. So this means in the open container, the cell present, but in the sealed one, there is no cell present here. So the no change in the pH and no change in the color. So the scientists belong to the germ theory say that this experiment proves that if there is no cell present in the bottle, that means no new cell arise, and the solution keep unchanged. But for the open container, the cell microbe, for example, from the air, can come inside, and then they dividing, and they make the new cell, and the more cell, the more change of colors in the solution. But the spontaneous theory, they didn't agree with that, because they say that the air contains a life force, and the life force necessary for the growth of spontaneously generated microbe. So if you seal the container, you will prevent the life force for the spontaneously generated microbe. So because of that, this experiment is failed. So that is the That is the way the spontaneous theories they say about the experiment, and they still disagree with the result of the experiment, and the two theory still make the argument for quite a long time. I think for nearly 1 century, they still not agree with each other about where do cells come from. Until the 1862, the year of 1862, when the French Academy of Sciences offered a prize to anyone who could solve this century-old problem. It seems very, how to say, now we can smile about that, but at this moment it's a serious problem about to prove where do cells come from, where do cells come from. And until the end of the 19th century, one scientist, he discovered the way to answer the question how, and also this scientist belonged to the germ theory, and he set up one type of experiment to prove that the cell come from pre-existing cell. And he set up this type of bottle to answer the question. His name is Louis Pasteur, very famous scientist involving in the microorganism. I think all of you already knew about Louis Pasteur, right? So he designed a specific bottle that he called the Swan's neck flask. And you see here, instead of the straight neck as normal, he made the neck like the swan neck, the swan's neck with the, how to say, with the side, not the, but with the neck is not straight as you see here. They make a like the curve in the neck of the bottle of the flask. So using this bottle or using this flask, he repeats the experiment. He repeats the experiment we already performed before. So here is the way that Louis Pasteur performed the experiment with the swan neck flask. Also, based on his experiment, you can know about how the study can perform in the real life. So whenever you want to do some like scientific research, so the first thing that you always have to have the question, the research question. Okay, so that means you have to you need to know about something. You also have the query about something. So you have to find out the way to answer this question. So always research question is the first thing appear and for the research. So in this case, the question is, do cells arise spontaneously from other cells? So that is the question for the experiment of Louis Pasteur. And he, based on his knowledge and based on the fact from the other scientists, there are two hypotheses. The first one, the spontaneous generation hypothesis, is cells arise spontaneously from non-living materials, like the life force in the air. And the second hypothesis is the cells are produced only when pre-existing cells grow and divide. So that is the two hypotheses can obtain from the experiment. So based on the result you receive, one hypothesis will be right and the other will be wrong. So he performed with the two types of the flask, the straight neck flask and the swan's neck flask, in the same condition. So I will not repeat about how he performed the experiment, but I just say about the result he got from his experiments. So after boiling and let the two bottle open, and he observed that in the straight naked flask, the color changes, but for the other flask, that means the swan's naked flask, the color is was not changed. So that means in the straight naked flask, there were cells present inside the solution, but for the swan naked flask, even the flask is open, but there is still no cell presented inside the solution. Why? Why the cell was not presented here? So the answer is come from the structure of the flask neck, because it is a swan's neck flask. That means they make a curve in the neck part here. So it's like the trap in this part of the neck. It appears like a trap of the bacteria or the microbe from the air. So when they heated the bottle and the water vapor, they come and they will be trapped at this part of the naked. So the microbe come from the air and also will be kept here in this part. So they stay in this part and they cannot come through this part to come inside to the bottle. So in this part, the color changes, but inside the bottle, the color doesn't change. So because the cell is trapped here and cannot come inside in this part of the bottle. So this result, they strongly support that with the pre-existing cell, so the new cell will be made, or the new cell come from pre-existing cells. But even with the open flask like that, the cell cannot come inside because they are trapped in this part. So the color in this part doesn't change or didn't change. So because of that, they strongly so that the hypothesis of the spontaneous generation is failed, or was failed. Sorry. So the cell arise only from pre-existing cell. So that is the experiment of Louis Pasteur to prove about the germ cell theory or the germ cell hypothesis. Do you understand the experiment?
Yes.
Okay.
Yes.
Okay, good. So that is the way the experiment is performed and the way we do the research. You have the question, so based on your notice, you will set up the experiment to find out the answer. So based on the answer, you can give the conclusion of your hypothesis is right or wrong. So in the near future, whenever you come to the lab and you want to do some kind of research, and you will see that all of your research will also do by this way. So Louis Pasteur gets the prize, and he also support for the third statement of the cell theory that all cells arise only from pre-existing cell. So that is the three statement for the classical cell theory in the 18th and 19th century. And here are the three scientists who made up the classical theory. Theodore Schwann, he was made up the first statement. He made up the first statement. The cell is the basic unit of life. The second one, Matthias Jakob Sladen, and he gives a statement of all organisms are made of cells. And the third one is Rudolf Ludwig Kahn. He states that all cells arise only from pre-existing cells. And Rudolf also is a pathological scientist. He worked on the cell a lot, and he also the scientist who first discovered that the cancer comes from the cell. The nature of the cancer comes from the cell. Of course, with the time, with the development of the technique and the more knowledge is gained, so the more understand or the more thing we know about the cell, and the statement of the cell theory will increase. So now the modern version of cell theory includes extra information. So what are they? Of the new statement in the modern cell theory. So for the modern theory, they state that energy flow occurs within cells, so involving in the energy flow. So we know that on the cell, for maintenance of their life, they need energies. So the energy can be made from the cell and used by the cells. The second statement is involving in the DNA molecule. The hereditary information, that means the DNA, is passed from cell to cell. And the third, involving in the basic chemical composition, that all the cells have the same basic chemical composition. So it's the three more statements added on the cell theory for now. And of course, I think in the future, more statements also will be added on the cell theory with the more knowledge we gain or we got. And maybe some old statements will be deleted from the cell theory with the new knowledge we got. So I already mentioned that based on the limitation of the knowledge or the technique, so the scientists at the 18th century or 19th century, they did not recognize or understood about the cell division or the other cell structure. And one of the important technique involving in the cell discovery is the microscope technique. So the discovery in the cell biology would have been possible without the invention of the microscope. And for now, we have many modern techniques involving in microscope, like scanning electron microscope, in the software we call SEM, and transmission electron microscope, TEM. This is the highest technique of the microscope for now. And with this instrument, you can observe the small molecules stay inside the cell with the nanometer range. So with the normal or the light microscope, we can observe only the micrometer range. But for the electron microscope, you can observe something below the micrometer size and at the nanometer size. In the cell, there is some structure. They have the micrometer size, but the other structure, they have nanometer size. So we call the micrometer scale and nanometer scale. For the micrometer scale, you can count with the organelles such as mitochondria or nucleus. The nucleus is the most or the biggest organelle inside the cell. The chloroplast also quite a big organelle that you can observe through the light microscope and also belong to the micrometer scale. The other structures such as ribosome, the DNA, or the cytoskeleton, such as the microtubule or the actin filaments, you cannot observe through the light microscope or as the micrometer scale. You have to observe this structure through the TEM or SEM microscope. That means the electron microscope, because the size of the structure is quite small as the nanometer scale. For example, the microtubule you see here, the diameter of one microtubule is about 25 nanometer. For the actin filament, it even smaller, with the 7 nanometer in the diameter. And the DNA, only 2 nanometer for the DNA helix between the two strands of the DNA. So this only after the electron microscope is discovered or is invented, they discover the DNA and the RNA molecule inside the cell. So microscope technique is very important for the cell research. And whenever you want to do the research on cell, you have to know about the microscope technique. Here's the example of the microscopy and the visualization in order to help you to understand more about how the microscopes can help you to observe the structure in the living things. For example, you can observe some small thing by your naked eye with the smallest distance of millimeter. Okay, so now use your eyes and try to recognize the distance between the 2 points and figure out how smallest of the distance you can observe for each of you. Try to figure out what is the smallest point you can recognize between, what is the smallest distance you can recognize between the 2 points. Have you ever tested this capacity of your eyes? Okay, or maybe another way to ask, what is the smallest distance that the naked eye can recognize between the 2 points?
I think it's about 20 millimeters.
Oh, I think it's not so sharp eyes. 20 millimeter?
2 millimeters, I think.
2 millimeters. I think you already use a ruler, right? So can you recognize the point, the 2 point of 1 millimeter? I think
No, I
you
think
can
I can't.
do.
No.
You can't.
Yeah.
The other?
I think we can recognize about 0.1 millimeter.
Yeah, good. I think that is a more suitable distance than the 1 millimeter or 2 millimeter only. Because you can easily to recognize the 1 millimeter on the ruler you use, right? Whenever you want to draw something. But you can also even look in more detail. That means you can recognize something smaller than the 1 millimeter. And the shortest distance that we can recognize is about 0.1 millimeter. Just look in your hand. Okay, you can recognize some line on the tip of your hand, right? Of your finger. And normally the distance between the two lines is about 0.1 millimeter, and you can recognize that. So by look at this slide. So by your naked eye, you can recognize about the micrometer, but about the millimeter. So the smallest is about 0.1 or 0.2 millimeter by the naked eye. And below this distance, you cannot recognize, right? And you need the support from the light microscope. So for the light microscope, you can observe something in the micrometer range or micrometer scale, such as the layer of your skin cell, or some structure inside the cell, some big structure inside the cell, like mitochondria or the cell nuclei, as I mentioned before. And all of these structures, they are at the micrometer scale. But for the other structure or organelle which is smaller than micrometer, I mean the 1 micrometer, below the 1 micrometer or below the 1.2 or 0.2 micrometer, so you cannot observe through the large microscope. And as that, as this size, you need the support from the TEM, that means the electron microscope, to observe them. So from the cell, so to the organelles and the molecule and the atom, they are very different in the size. They are big different inside with the size, and based on the size, you need 7 types of supporting from the microscope technique to observe this object. So with the electron microscope, even you can observe the atom. That means some very small object in the life. So for the microscope technique, also you have 7 type of laser light microscopy to observe the cell. So you can see here with the different mechanism you use on the light microscope, you can observe different things inside the cell. The cell can be stained or unstained. That means you can use some dye to specifically color some specific structure inside the cell, and based on the difference in the color, you can recognize different structure inside the cell. For the fluorescent microscope, you can use some fluorescent probe, like the antibody or some DNA sequence, RNA sequence. So you can color some structure or some organelle or some protein or some parts of the DNA inside the cell, and based on the signal of the fluorescence, you can know where and how the molecule is present inside the cell. Of course, if you want to do the research on the cell, so you have to understand about this technique. Here is some example about how the cell look or how the object look like under the electron microscopy. So these 2 images was taken by the SEM. That means the SEM. That means the scanning electron microscope. So the electron microscope was invented in 1932 in Germany, and came into widespread biological use in the early 1950s. So as I say, there are two basic designs, the TEM and the SEM. So for the scanning electroscope, you can observe the surface of the object. That means the surface of the cell, or here, like the surface of the pollen grain. In the left image is the surface of the human neuroblastoma cell. So neuroblastoma, that means one type of the cancer cell in the neural system. So you can see the surface of the cell they also have some like the hairy surface in the outer membrane of the cell with the different size and morphologies here of the cell. And in the right image, so you can see the surface of the pollen grain with many homologous structure in the outer part of the grain. For the TEM image, the TEM image will help you to look inside the object, look through the object. That means the structure inside the thing you want to observe. For example, here, you can compare the SEM image and the TEM image of the podocyte. This is one type of cell in the Bowman structure of the kidney, in the kidney. So from the same image, you can see the podocyte have the thin morphologies, like the filament morphology of the cell. But when you look through the TEM image, you can recognize the cell nucleus, the cell nucleus of these podocytes, of the two agents of podocyte here. And the other structure inside the cell, you can also observe through the TEM image. So that is the two different in the SEM and the TEM image when you use this technique to observe the cell. Okay, so through the histological discovery of the cell, you also knew that there are 7 types of the cell, and they can divide into two main types of cells. That is the prokaryotic cell and the eukaryotic cell. So prokaryotic cell, like the bacteria or the algae, right? And for the prokaryotic cells, they are very simple. That means the structure or the organelle inside the cell is very small. I think it's the most simplest life in the earth. So this is the structure, the drawing of the main structure in the prokaryotic cell. So you can see that they contain only the cytoplasm with only one type organelle, ribosome. So ribosome is the only organelle present in the prokaryotic cell. Even the nucleus, they don't have the nucleus, they have only the nucleoid. That means the nucleus zone. That means the area they contain the DNA or RNA molecule inside the DNA of the cell. Also, surrounded of the cell, they have the cell membrane, also the cell wall. They have in some type of cell like E. coli, they have the outer membrane and the inner membrane, and a cell wall between the two membrane. And the other cell, they have the flagella, or they have some filler surrounding the cell. For the eukaryotic cell, they are much more complicated with much more organelle inside the cell. So for the eukaryotic cell, we have the animal cell and the plant cell. They also share some common organelles and also different organelles. And in the cell biology course, you mostly focus on the animal cell because you also study about the plant cell in the other course. So for that reason, in this course we mostly concentrated on the animal cell. Okay, so now we have question and also new practice to do the assignment in the classroom. Okay, so you through the introduction part of cell biology, you may know about what is the life, right? What is the life? The definition of life, and life is easy to recognize but difficult to define. So I think it's difficult to define, right? Because we have many ways to define what is the life, and we, as I said to you about the biological ways, life is a thing that contain or made up of cells. So according to one popular biology text, living things are made from these 7 characters. So based on these 7 characters, score a person, a vacuum cleaner, and a potato with respect to the characteristics. That means a person, a vacuum cleaner, or potato, how many characters they have based on these characters listed here. So please do it on your Google Classroom. Okay, I will assign this, and you have 5 minutes to complete your assignment.